Hello, BISC130. This is the beginning of recorded lecture 1-4. We are still in the biological macromolecules, chapter 3. We talked about carbohydrates and lipids in the last one. Now we're going to pick up and talk about proteins. So for our other macromolecules, I started by talking about structure, and then we talked about function. For proteins, I'm going to go in the opposite direction and start by talking about their functions, because there are a lot. Proteins, compared to all the other macromolecules in this chapter, are capable of doing a ton of different jobs. So I'm going to list some of these, but you should know, as long as this is not an exhaustive list. Proteins do so many things. So one of their functions, as an example, is enzymes. So yeah, here's a figure from a later chapter. We'll get into this in a later in this quarter, but yeah, there are many proteins that can help make or break chemical bonds. Very important job. But that's not all. Proteins can also be involved in defense. Oh, here's a slide from BISC132, the other class I may teach, depending on what your major is. Antibodies, part of the immune system. These Y-shaped things are proteins, very important part of immunity. So proteins can also be involved in defense. Proteins can also be involved in transport. They can carry fats and other things in the bloodstream. Here's an example of a protein that carries oxygen through your bloodstream. Very important in getting that oxygen from the lungs to everywhere else in the body where it needs to go. Proteins can also be involved in structural integrity. So we saw polysaccharides, carbohydrates that were involved in structural integrity in the previous lecture, but there are proteins that can do a similar thing. There are proteins that give strength to your cartilage and to your fingernails and toenails. Proteins can be very strong. Proteins can also be involved in muscle contraction. Again, another slide I stole from my BISC132 lectures. But yeah, the way your muscles work involves proteins working off of one another. So proteins can be involved in this as well. And finally, again, just for this list, proteins can be involved in regulation. I defined hormones in the key terms in the previous recorded lecture. Steroids were examples. Some steroids were examples of hormones, these signaling molecules. But proteins can be hormones as well. So some hormones are not steroids. Some hormones like insulin, the process shown here, insulin and glucagon are proteins that serve as these sorts of signaling molecules. So with all these different very diverse functions that proteins can have, the question should arise, how can one type of thing, how can one class of macromolecule be responsible for all these different, very different jobs? And the answer has to do with how these things are built. Proteins are, like carbohydrates, polymers. But instead of being polymers of individual sugars, proteins are polymers of what are called amino acids. Oftentimes abbreviated just as lowercase a period, lowercase a period. But yeah, proteins are polymers of amino acids. So what do these amino acids look like? Well, this is part of what gives proteins their magic touch of being able to do all these fun things. Amino acids come in all shapes and sizes. So do not memorize this table, but yeah, these are the 20 commonly used amino acids. Some are short, some are long, some are non-polar, hydrophobic functional groups, some are polar, some are positively charged, some are negatively charged, some have big bulky rings, some are more, straight linear, some have weird elements like sulfur as part of their functional groups. So these are really diverse building blocks. That's how we can have a, functionally diverse macromolecule doing all these different things, because its building blocks are incredibly diverse. And again, it's a polymer of these things. A protein is just a bunch of amino acids strung together to form a larger structure. And again, you don't need to memorize all the structures or names, but you should know there are 20 of them, 20 commonly occurring amino acids. And they look different and they do different things. There are different functional groups on these amino acids, giving them different chemical properties. It is worth pointing out most proteins, at least overall, are hydrophilic. Most proteins will dissolve in water. There are some hydrophobic amino acids, but on the whole, most proteins have enough of the hydrophilic ones to where most proteins will dissolve in water. So in our last polymer, carbohydrates, we had a special type of bond that connected the individual units together. That was a glycosidic bond when we were talking about carbohydrates. We have a similar thing here when we're talking about connecting amino acids. Amino acids are linked by a type of covalent bond called a peptide bond. And yep, there's an amino acid, there's another amino acid, here's the peptide bond connecting those together. Again, it's just a covalent bond, but it's a special type called a peptide bond. For this reason, an alias of proteins, another name that proteins go by sometimes, is polypeptides, which should make sense. They are polymers of things connected by peptide bonds. So it doesn't roll off the tongue as well as protein, but polypeptides and proteins are the same thing. Now, proteins have very complicated structures. Sometimes they're drawn, very cartoony. Sometimes they're drawn more realistically. Obviously, there's a lot of very specific shape that goes into any protein. In order to get this shape, the protein has to fold up in a very specific, complicated way. And to describe this structure, we have 4 distinct levels. So there are 4 levels of protein structure. The first level, appropriately named, is called primary structure. The primary structure of a protein is just the sequence of amino acids. So yeah, the key terms say primary structure is the linear sequence of amino acids in a protein. So yeah, you can describe the primary structure just by listing off, glycine, isoleucine, valine, glutamate, glutamine, cysteine, alanine, serine, valine. Just list off the order of amino acids, and that's the primary structure. Obviously, it doesn't stop there. There's more complicated structure to this, but yeah, primary structure is just the sequence of amino acids. Next, we have, again, appropriately named, secondary structure. Secondary structure are, to read from the key terms, regular structures that proteins form by intramolecular hydrogen bonding. These secondary structures, they're not the full shape. These secondary structures are common patterns, common structural motifs. Before we see this, let's take a look at these. So this is something called a beta sheet or a beta pleated sheet. You have a line of amino acids, a bit of a loop, and another line of amino acids, and these two ribbons are held together by hydrogen bonds. You could have a beta sheet with 2 sheets, you could do 3, you could do 4, you could do more, but this type of interaction, just having a sheet and another sheet, is an example of secondary structure, very common in proteins. The other common secondary structural element is the alpha helix, where, line of amino acids here, but it sort of winds into a sort of spiral staircase or helix structure. And if we look closely at the interactions here, it's hydrogen bonds that is holding this together. Yeah, we talked about hydrogen bonds a while ago, and I said they were the weakest of the 3 chemical bonds we talked about, but there are just so many of these hydrogen bonds across a beta sheet or within an alpha helix. These tend to be pretty durable structures just because there's so many of these hydrogen bonds. So to summarize, these secondary structural elements, these common patterns, include the alpha helix and beta sheets, importantly held together by hydrogen bonds or H bonds. So again, this is not the full 3-dimensional structure, it's just these common patterns. If we want the full 3-dimensional structure, we go to, predictably, what is called tertiary structure. The key terms define tertiary structure as a protein's 3-dimensional conformation, including interactions between the secondary structural elements. So what holds this thing together? Again, this is the full 3-dimensional structure. We could see this shown in different ways. You have this very popular ribbon diagram. Oh, there's an alpha helix, there's 2 beta sheets together, some unstructured loop regions here, but yeah, we see secondary structural elements. This is one way to draw tertiary structure. Here's a space filling model that's just another way to draw this. I think this is probably the more common way to see this, the ribbon in textbooks. But yeah, these are just different ways of showing tertiary structure. So what holds this thing together? Well, like this says, it's held together by interactions, but it's a little bit more than that. So if we look at what actually holds together this complicated three-dimensional shape, we see a lot of familiar things. So remember, some of these amino acids have negative charges, and some of these amino acids have positive charges. Guess what happens between positive and negative? They're going to want to get together. They're going to want to form an ionic bond. So ionic bonds hold together this tertiary structure. So do hydrogen bonds. It's not just within the beta sheets or alpha helices. These distant elements can interact, and by hydrogen bonds, that holds together this overall shape as well. We also have, if you're dealing with those special sulfur-containing amino acids, special connections called disulfide linkages. So, yeah, it's not really showing the beads on the string here, but yeah, there's an amino acid here, an amino acid here, an amino acid here, an amino acid here. If you've got two sulfur-containing amino acids, even if they're far apart from one another on the string, this disulfide bridge or disulfide linkage is a covalent bond that links these two distant parts of the protein together. And finally, there's a new type of interaction, something called hydrophobic interactions or hydrophobic exclusion. So remember, some of these amino acids were hydrophilic. They want to be facing the outside world. They want to be touching water. But plenty of these amino acids are hydrophobic and don't want anything to do with water at all. Well, these hydrophobic amino acids help the protein maintain its overall three-dimensional shape because these hydrophobic amino acids will fold in such a way that they are in the core of the protein. They're hanging out with one another. They're touching one another. They're not touching water. So this sort of mutual dislike of water that puts all of these things together in the center or core of the protein is another thing that holds together this structure. So this is a great visual for these things, but if you want this listed out, tertiary structure is held together by interactions including hydrogen bonds, ionic bonds, disulfide linkages. Again, this is a type of covalent bond, not the same thing as a peptide bond, just a different type of covalent bond that's very specific here in the tertiary structure. And then this hydrophobic exclusion, where hydrophobic amino acids fold into the core of the protein. Now, if you have a good memory, you may remember I said there were 4 levels of protein structure, and this feels like it should be the end, right? It's the full 3-dimensional structure of the thing. How could you possibly get more complicated than that? Well, you can, in some cases, have what is called, again, predictably, it's a lot of key terms, but they all have pretty easy names. Level 4, quaternary structure, which the key terms defines as the association of multiple polypeptide subunits in a protein. In other words, quaternary structure is when you have multiple protein units. Each one has its beginning and end. Each one of these has, its own distinct string of beads and its own tertiary structure. Quaternary structure is when you have multiple things coming together to form a functional protein. Here's that hemoglobin protein responsible for carrying oxygen in your red blood cells throughout your bloodstream. It has a quaternary structure. It's got 4 distinct polypeptide subunits that come together to form functional hemoglobin. Importantly, this is optional. So the quaternary structure exists for some things, but not everything has this. This lysozyme, which I showed a minute ago, it does not have quaternary structure. It does its job. By the way, this breaks down bacterial cell wall. You've got this in your eyes, for example, to prevent bacterial infections in your eye. You've got this enzyme that breaks apart the bacteria cell wall. This doesn't have any quaternary structure at all. So this is definitely optional. Not all proteins have quaternary structure, but we're going to see proteins later in the quarter that have quite complicated quaternary structure. So that's our 4th level of organization in putting these proteins together. Now, for a protein, its 3-dimensional shape is absolutely key to its function. This lysozyme is only able to bind to bacterial cell wall and break it apart. It's only able to do its job because it has this very specific pocket here, this very specific curvature. Everything about its structure is very important for it to do its job. You know, whether it's breaking down cell wall or carrying oxygen for proteins, shape is everything. So that means if you lose shape, you're going to lose everything. You're going to lose the ability to do whatever it is you need to do. Protein denaturation, or sometimes it's just called denaturation, is defined in the key terms as the loss of shape in a protein. And so, yeah, protein denaturation is when you go from this properly folded, all these complicated structures here, secondary structural elements, properly folded protein, to just a string of amino acids, losing this complicated structure. So what could cause this structure to fall apart? Well, this is actually something you're pretty familiar with, whether you know it or not. You can easily denature proteins through heat. So yeah, if you've ever cooked an egg before, egg has a lot of protein in it, especially in the white. If you heat an egg up, it is going to denature those proteins, and you can visibly watch them go from clear to white as these proteins lose their shape and they lose their function. We don't necessarily care about, making them lose their function. We do care because we don't want bacteria inside of in this egg causing disease. If we denature the bacterial proteins, that's going to make them dead, and that's going to make this safer to eat. So heat is one way to denature proteins. Hey, if there are any amateur cooks listening, there are other ways to make food safe to eat. Heat is, definitely the most common way to do this, but another way that will denature proteins is acid. If you've ever had ceviche before, this is fish or other seafood that has been made safe to eat through the application of lime juice or lemon juice or some sort of acid. So a low pH or high pH can also cause protein denaturation. And here's kind of an obscure one, but another one from the world of food here is messing with salt levels. So messing with salt content to like salt cure meats, that can also cause proteins to denature. So yeah, all three of these things can contribute to protein denaturation. Changes in temperature, changes in pH, changes in salt content. And this leads to loss of function. Protein can't do its thing if it doesn't have its shape. This is usually irreversible. If you take this cooked egg and pop it back in the cold refrigerator again, it's not going to turn raw again. It's going to stay cooked. This is a no take backsies thing, which makes it all the more scary. You absolutely do not want your proteins becoming denatured. So that's why, once again, we come back to homeostasis, preventing denaturation. We regulate our temperature or pH or salt content in our cells and the fluid around our cells. We don't want this to happen to us. Okay, we've done carbohydrates, we've done lipids, we've done proteins. Last biological macromolecule, or should I say molecules. This last one is going to be two that are kind of put together. To end out this chapter, we are going to talk about deoxyribonucleic acid and ribonucleic acid, more commonly known as DNA and RNA. These things are, once again, polymers. So polysaccharides were polymers, proteins are polymers, DNA and RNA are polymers as well. So that means they're a big structure made of a bunch of smaller things linked together. In this case, DNA and RNA are both polymers of things called nucleotides. So actually, before we talk about DNA and RNA at all, we have to talk about their building blocks. We have to talk about what nucleotides are. So nucleotide structure, again, don't have to memorize how to draw this perfectly, but there are, color-coded for your convenience here, 3 important structural components to a nucleotide. So, First up, something that should look familiar to you. This, this ring structure here. This is a carbohydrate. This is a 5-carbon sugar. So yeah, nucleotide structure consists of a 5-carbon sugar. So DNA and RNA are both polymers of nucleotides. They both use these things. There are a couple of small differences between the RNA building blocks and the DNA building blocks. And here is one of them. The 5-carbon sugar used by RNA is a sugar called ribose. The 5-carbon sugar used in the nucleotides of DNA is called deoxyribose. It's the difference in this one little position right here. How are you going to remember which one is which? It's right there in the name. RNA is ribonucleic acid because it has ribose. DNA is deoxyribonucleic acid because it uses deoxyribose. So this is a slight difference between the two building blocks, but it's easy to remember because it's right there in their names. Okay, so we've got our 5-carbon sugar. We also have something else that should look familiar to you, the phosphate group. So we saw phosphate groups earlier when we were talking about phospholipids. Same thing that was true there is true here. The phosphate group has a negative charge to it, which is going to make these nucleotides hydrophilic. It's going to make them negative. It's going to make them water loving. It's going to make them charged. Importantly, this phosphate group is attached to the sugar, ribose or deoxyribose, at a very specific location. It's attached to the 5th carbon. So biochemists like to number the carbons, so they can be very precise when they talk about things. So we would call this carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. But oops, we can't call these carbon 1, 2, 3, 4, 5 because we need to number the carbons over here in this third component, which we'll get to in just a minute. So instead of calling this carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, we have to call this carbon 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. The phosphate group is attached very specifically to the 5 prime carbon of this 5-carbon sugar. For this reason, it is often called the 5 prime phosphate, referring to which carbon it is attached to. Might seem like too fiddly of terminology to get into. Trust me, when we talk about DNA structure and function and how this stuff works, the numbers and the positioning and the terminology here will be important. So we got a phosphate group called the 5' phosphate attached to the 5' carbon of the sugar. And finally, we have up here the nitrogenous base. Now, here's where it gets a little complicated because there are several different nitrogenous bases that could fill this spot in the nucleotide. In fact, there are 5 possible bases that could be slotted in up here. This is just showing one of them. The 5 nitrogenous bases, and again, don't memorize their structures, but you do need to know their names. They are adenine, guanine, thymine, cytosine, and uracil. Uracil obviously seems like an odd one out just based on its name. All the rest of the ones end in "ine." Uracil doesn't. And that's an easy way to remember that uracil is actually weird. Uracil is only used in RNA nucleotides. You will not find uracil in DNA. In contrast, thymine is only used in DNA. You will not find thymine as a building block, as a nitrogenous base in RNA. The other 3, adenine, guanine, and cytosine, they're found in both. You'll find adenine, guanine, and cytosine in both DNA and RNA, but these two are our exclusives. Thymine's only in DNA, uracil is only in RNA. Now, the other thing that you will notice, again, without memorizing the exact structure here, is that there are 2 basic categories of these nitrogenous bases. big ones and small ones. Ones that have 2 rings as part of their structure, and ones that have 1 ring as part of their structure. There's some terminology here, which again, trust me, will be important when we talk about how these things actually work. So I'm not just making you memorize fiddly stuff for no reason. The big ones, adenine and guanine, are called purines. The smaller ones with only 1 ring, thymine, cytosine, and uracil, are pyrimidines. So A and G are purines, 2 rings. CTU are pyrimidines with 1 ring. Just commit this to memory. This will come up. Now, just like any other polymer, we need to link a bunch of these individual units together into a big long string of things. We saw this with polysaccharides, we saw this with proteins, we're seeing this here in DNA and RNA. And just like we had a special name for the bond that held together the polymer for those other things, we got a special thing here as well. It's called a phosphodiester bond. So in a big string of nucleotides, they are linked together by a phosphodiester bond. Again, don't get this mixed up with our other polymers. It was the glycosidic bond that did carbohydrates, sugars. It was peptide bond that did proteins. And now we have phosphodiester bond that does nucleotides. This one's easy enough to remember, I think, because it's got phospho in the name, and this bond involves the phosphate group of the nucleotide. So this isn't just connecting the nucleotides together, it's connecting them at very specific positions. So, oh yeah, we've got our 1', 2', 3', 4', 5'. We got it labeled in this figure. So yeah, here's our 5' carbon, there's the phosphate, the 5' phosphate, and we can see where it connects on the next nucleotide. It doesn't connect to 1, doesn't connect to 2, doesn't connect to 4, it specifically connects to this 3' OH group. So the phosphodiester bond is from the 5' phosphate of one nucleotide to the 3' OH of the next nucleotide. I've got that written here. Nucleotides are connected by a type of covalent bond called a phosphodiester bond. It's the 5' phosphate of one connected to the 3' OH of the next. And yeah, this is just showing 3 nucleotides. But yeah, DNA and RNA get to be pretty long. So you could have many nucleotides all connected by lots and lots of phosphodiester bonds into one big long strand. Actually, I say one big long strand, but I think most of us are familiar with DNA and its very unique and cool looking structure, that it's not one strand, it's 2. So DNA consists of 2 strands of nucleotides connected by phosphodiester bonds that have come together to form this double helix structure. So DNA is a double helix formed from 2 strands of nucleotides. What holds this structure together? Well, we know that phosphodiester bonds hold together each individual string, but what force is holding these 2 strings to one another? Well, if you squint hard, you can see some little black dots here. If we zoom in, we can see it's hydrogen bonds. Once again, hydrogen bonds, the weakest of the chemical bonds we talked about, but there are just so many of them. Every single pair of nucleotides has several of these things. So yeah, this makes a very stable double helix because of hydrogen bonds between these nitrogenous bases on the nucleotides. And here we see an example of how the pairing up, the partnering between nucleotide on one strand and nucleotide on the other strand, nucleotide on one strand, nucleotide on the other strand, is not random. It is very specific. These 2 strands do not have the exact same sequence, but they do have what is called a complementary sequence. These 2 strands will have nucleotides that partner up in such a way to have the strongest connection possible. These are the strongest possible combinations of nucleotides. Thymines, ideal partner is adenine. Just if you tried to partner thymine up with guanine, it just wouldn't have as strong of a connection. If you tried to pair thymine up with cytosine or even with another thymine, it wouldn't hold together well. The best partner for thymine is adenine. The best partner for adenine is thymine. The best partner for guanine is cytosine. The best partner for cytosine is guanine. So here's my illustration here. These two strands pair with one another in order to optimize hydrogen bond formation. That's why these rules exist. These rules, C to G and A to T, I should make this in an extra large font because this is an extra large concept. These base pairing rules are very important. It's how you partner these things up in order to have the strongest hydrogen bonds possible. And, oh, hey, using this terminology here, purines and pyrimidines, you'll notice that these always partner up to where you have a purine and a pyrimidine. A purine and a pyrimidine. If you had two purines, two of the big ones, they would be too smushed together. If you had two of the smaller ones paired against one another, they wouldn't be able to reach. So these ideal partnerships, A to T, T to A, G to C, C to G, these involve a purine paired with a pyrimidine. So let's walk through an example of this. So, oh yeah, here it is in much bigger font. These are the rules, C to G, A to T. And if you want a dumb way of remembering this, to me, capital C and capital G, these two letters they're drawn in a very similar way. So in my mind, at least, it's just easy to remember that C and G go together because the two letters just so physically resemble one another. And if you remember that C partners with G, then the other two obviously partner with one another. So what this means is if you have a sequence of DNA here, don't memorize this, I just made this up. CCTAGCAAG. If this was one strand of the DNA double helix, knowing these rules, you should be able to figure out what the other strand would be. So if one strand has C, the other strand should have G right across from it. The next position should be another G, C pairs with G. The next one should be A. T pairs with A. Then there should be a T, then C, then G, then T, then another T, then C, and yeah, repeat this millions of times if you want the authentic experience of building a DNA double helix. But yeah, this is what I mean. The two strands of DNA are not identical in their sequence. They are complementary in their sequences according to these rules. So this optimal pairing results in what are called complementary sequences between these two strands. And again, this is a great sort of overview figure showing these hydrogen bonds and showing the thymine, adenine, guanine, cytosine. Now, all this was DNA. What about RNA? Well, luckily, RNA is just single-stranded. Most of the time. So we don't have to deal with any of this type of interaction with RNA. There's a DNA double helix here, but RNA, most of the time, is just single-stranded. And I also like this figure because, yeah, color-coded, it shows, here's that uracil, the RNA exclusive nitrogenous base. And yeah, we see some of this pinkish purple only in RNA, but not in DNA. And there's our thymine, blue here, only in DNA, none of this blue thymine in RNA. But yeah, DNA is double-stranded, RNA is not. So that's the structure. What about the function? Well, because of this DNA, because of this double helix structure, DNA is a very stable molecule. It has a lot of strength holding it together because of all these interactions in this double helix. The function of DNA is unlike the function of anything we've seen in this chapter. It's not storing energy or being structural integrity. The job of DNA is to store information. So You'll appreciate this more later in the quarter because we will talk about how this stored information actually gets used to do stuff in the cell. But for now, I'll just say that the job of DNA is like a blueprint. It's the instructions for how the cell is supposed to build proteins and conduct business and respond to things. This is the code. This is the genetic information. This is the blueprint. So long-term storage of these important instructions for how to sell. RNA, in contrast, is not quite as stable, but its job is oftentimes the same thing. A lot of RNA has the same job of being information, of being a blueprint. Most RNA functions as information, but it's a short-term copy of that information. So if this is the master blueprint for how to build a house, RNA is going to be the photocopy of that blueprint that you take to the construction site. And this is going to get dropped in the mud, and this is going to get torn, and this is going to get scratched up or whatever. You crumple this up, you throw it away, you make a new copy. You keep the DNA safe because that's your master copy of all this very important information. The RNA is just a short-term copy of that. But at the end of the day, these are both just instructions. These are both just information. So most RNA functions as a short-term copy of DNA's information. It's still an information molecule. However, it is worth pointing out here, and we will see this in action later in the quarter, there are actually some RNAs that have structural or functional roles. There are going to be some RNAs that are actually enzymes that make chemical reactions happen. There are going to be some RNAs that support the structures of things that are used to physically build stuff. And again, we'll see those when we get to them. I just want to note that some RNAs are weird, but most RNAs are a copy of DNA's blueprint information. So with that, we come to the end of the biological macromolecules next time. So this is the end of recorded lecture 1-4.